So it's my great pleasure to introduce today's first speaker, Apoorva Baluapuri, a postdoc in Karen Edelman's lab at Harvard Medical School. So Apoorva completed his PhD at University of Würzburg in Germany, where he has been focusing on the oncogene MYC. And his PhD was divided into several pieces, but importantly, he made two important discoveries where first he has uncovered how MYC selectively upregulate long genes by modulating RNA polymerase II dynamics through SPD5 and SPD6. The second is that he discovered how MYC multimerization helps cells to recover from replication stress by limiting checkpoint signaling. So he has been extremely involved in mentorship as well as teaching, and his research as well as his mentorship has kind of like experience has earned him many fellowships and awards, including the Outstanding Tutoring and Mentorship Award, the Federal German Excellence Initiative Fellowship, and the Harvard Medical School Intramural Fellowship. And in his postdoc with Karen, Apoorva has been investigating how loss of the integrated complex contributes to the neurodegenerative disorders, a very exciting story that he will be sharing with us today. Please join me in welcoming Apoorva.
Thanks a lot, Sky, for this nice introduction. I'm very excited to present this story and be a part of the Fragile Nucleosome series for the second time. I would be presenting this story, which recently came online. I hope you guys have a good, you like it. And let me start with a quick introduction to the transcription process. As many of you know, the eukaryotic gene expression is regulated at several distinct steps, with the key decisions being made during the early elongation part of it, meaning that after the RNA polymerase II initiates RNA synthesis, it pauses in the promoter proximal region. And this pausing is stabilized by two factors, SPD5 and the NEL complex. This is a fairly stable complex, and the complex remains stable until the arrival of the kinase PDEFB, which phosphorylates three things. It phosphorylates the C-terminal domain and the linker of the RNA polymerase II. It phosphorylates SPD5 and the NELF complex, which then dissociates with this from the polymerase, and this allows the binding of elongation factors to the RNA polymerase, and then the polymerase becomes fully competent to elongate productively to the end of the genes. And it's very important that the polymerase goes to the end of the gene and not terminates and resists the temptation to terminate in the middle of these long AT-rich introns, particularly at these things called cryptic termination sites, because this would become a problem for the cell. When this cryptic termination happens, what happens as a result is that it results in the retention of introns, and these retained introns can then be inadvertently exported to the cytoplasm. Now, this is a very bad news for cell because this has implications for both cancer and immune surveillance, since these aberrant transcripts can encode for anomalous or even oncogenic proteins. And moreover, our introns are so large and expanding in humans because they have been continuously invaded by repeat elements, which cause a lot of problems if they make out into the if they get out of the nucleus into the cytoplasm, you know. So what I'm trying to emphasize here is that it's clear that the pause elongation complex can be phosphorylated by PDFB and can ensure incomplete transcription across the genes. And this is good because otherwise it would be bad news for the cell. But what is less clear is that what happens if this kinase, PDEFB, never arrives to the promoters. When this kinase is not recruited to the promoter and the polymerase tries to sneak, the unlicensed polymerase tries to sneak, what happens? Research in the last 5 years has brought to light that this important job of ensuring that this unlicensed polymerase does not enter the gene body is done by a complex called the integrator complex. This is the termination complex, which, like the cleavage and polyrelation machinery, have both endonuclease activity to cleave the nascent RNA phosphatase activity and phosphatase subunit to antagonize the phosphorylation, thereby driving the transcription termination. Now, for those of you who haven't heard a lot about this complex, let me give a quick intro to that. This is a multi-subunit complex which was first discovered in the lab of Ramin Shekhatar, and it has about 15 subunits. All subunits except one are essential proteins. Depletion of them will kill the cell. However, there have been monoallelic loss of function mutations noted. That is to say, particularly in the subunits that I've highlighted here, these are associated with neurological disease when only one allele is mutated, resulting in a reduced amount of these subunits. I will just briefly visit one of such disorder because I'll come back to it. This is a disorder coming from INDES8 and 1 mutation. What happens in this disorder is the patients do not develop their cerebellum completely. The cerebellar part is hypocellular, filled with CSF, and this results in very massive neurological defects. A similar case was noted in patients with INDES11 mutation. And this was even more severe because the MRI I'm showing you is actually post-mortem. So the take home here is that the integrated deficiencies cause these massive defects by failures in migration or cell proliferation. And the question that I wanted to answer during my postdoc with Karen was that how does integrated deficiency contribute to these diseases? Some evidence and some help was already there. Before I joined Karen's lab, a lot of labs had worked on this, and including Karen's lab had found that it doesn't matter if you deplete the phosphatase subunit or the NB nucleus subunit in human cells or mouse cells or Drosophila cells, the same set of stress responsive genes are activated when integrator is depleted. This has been also shown by many other labs around the world. But I did not want to study the endonuclease or the phosphatase activity specifically. I wanted to study integrator here as a whole. So what we did was we collaborated with the lab of Eric Wagner, and we tagged the INTES1 subunit of this complex. Now, this is the subunit that forms a backbone and connects the whole complex to the phosphopolymerase. So what we thought was if we deplete this protein, the whole complex should fall apart. And as you can see here, now in these HEC293T degron tagged cells, if we add auxin, INTES1 completely goes away. And what I'm not showing you here is that the whole complex dissolves. The first question that I wanted to ask was, does this loss of the backbone subunit activate these stress responsive genes that have been traditionally seen? So what I did was I utilized a technique called TD-seq, which measures newly synthesized RNA via metabolic labeling of RNA. So when we perform a TD-seq, what we see is expected gene upregulation, including canonical integrated target genes, which are involved in stress response like FOS, as you can see here. And of course, what you see is many genes which are upregulated more than what they're downregulated, actually. These genes, as we have come to expect, are involved in the same ontology and functional categories that we have seen previously, including inflammatory and stress response. But the question that came up next was, does this nascent RNA change, does it get reflected all the way into the steady state RNA levels or not? So to answer that, I did yet another set of experiments where I did mature our total RNA-seq after 4 and 12 hours of INTES1 depletion. I collected the genes which were up and down regulated significantly in these experiments and performed clustering on them to find out which genes are upregulated earlier and later. The clustering looks like this. As you can see, there's a top cluster, cluster number 1, which is a set of genes that are only transiently activated. And these are the genes which are similar to Jun and FOS. So these are very early, these are immediate early genes which respond very quickly to integrator loss. But now what we can see is with better temporal control as the longest time points, which are also activated by sustained depletion of integrator. So in this cluster, what we see is not only the enrichment of genes which are the stress response genes, but we also see the genes which are the target of the transcription factors which were activated at an earlier time point. So integrator depletion increases Jun and phos level, they become proteins, and these proteins activate their own target genes. But what was a very interesting cluster was the cluster number 3, where we saw genes which only come up at the later time point. And if you look at these genes, of course, now there's a full spectrum of the stress response genes, but what we also see is the target of a transcription factor called ATF3. In fact, if we look at these genes, look at their promoters, there's a consensus motif of ATF transcription factor that comes out. And this was really interesting for us because ATF3, along with ATF4, is a key effector of something called the integrated stress response. Now, the integrated stress response is a very conserved pathway which consists of some stem cells which autophosphorylate themselves in presence of stress. This autophosphorylation reduces translation by phosphorylation of EIF2 alpha, thereby causing stress granule formation. And thus, but at the same time, there's few factors like ATF4 whose translation actually increases, and this helps in the mitigation of stress and restoration of homeostasis. If this does not happen, the cells will die out of apoptosis. And in fact, the diseases in which this happened, this chronic ISR activation, have been implicated in the similar kind of disorders which are caused by integrator mutation. So of course, we wanted to check, does integrator depletion causes expression of ISR effectors? And indeed, ATF3 is activated already at the RNA level. Its partner DD3 is also activated at the RNA level, but more importantly, both ATF3 and ATF4 increase in the protein level in a time-dependent fashion. The phosphorylation mark that activates it, that increases ATF4 level, that also increases. I'm not showing you in interest of time that the protein synthesis is inhibited as well, but what I will show you is if the stress granule formation happens or not. We measured stress granule formation by looking at G3BP1 immunofluorescence, and as you can see here, as the time passes by, there are more and more stress granule dots formed, and the even distribution goes away. What became obvious to us was that the ISR is definitely activated, but how was the question? And one thing that came out of this, one obvious schematic, that the possibility that comes from the schematic is that the RNA part is very obvious here. So the question was, does integrated loss allow for generation of double-stranded RNA or not? And this is not a very complicated experiment to do. There's an antibody which detects double-stranded RNA specifically, and immunofluorescence against this antibody shows that this double-stranded RNA starts increasing in the cytoplasm as integrator depletion time goes by. More importantly, the kinase that is involved, the sensor kinase, that also gets autophosphorylated. The PKR is the kinase which gets autophosphorylated. And more importantly, this PKR protein, the sensor, is in close proximity to the double-stranded RNA as observed by the proximity ligation assays in absence of integrator. The question next was, where is this double-stranded RNA coming from? How can transcription changes result in this? So to answer that, the PL experiments gave a kind of clue. What we could do was we could immunoprecipitate these RNA, sequence them, and perform peak calling to identify where they came from. And if you've been guessing that they're coming from repeat elements or not, you're absolutely right. But it was not a general repeat element, all repeat elements that we saw that were giving rise to double-stranded RNA. It was a specific category called signs. And these signs are interesting because they are subcategory. ALUs are typically in the euchromatin, not in the heterochromatin like the LTRs, and these are often in introns. So that made us ask that, are these actually coming from introns or not? And indeed, as you can see here, when we do this RIP-seq experiment, most of the peaks are coming from the introns. So what exactly is happening here? And I will answer that by one genome browser track. So what you can see here is the result of the immunoprecipitation of these RNA from the double-stranded RNA antibody and the PKR antibody that I have been saying as a sensor. What you can see here is that these peaks exist in kind of pairs. And if you look at the orientation of these repeat elements, they're actually existing in the inverted fashion, which means that this double-stranded RNA is coming from the same RNA molecule folding over itself. And second thing, what you can hope observe here is that there is this increased transcription in the beginning of the genes, but not towards the end of the gene or the middle of the genes in absence of integrator. And this means that in absence of integrator, the checkpoint or the gatekeeping is released. Unlicensed polymerase goes into the gene body, but it cannot transcribe very far. And this happens across all the genes that we had studied with PKR peaks. So the model here that I'm trying to explain is that in normal conditions, integrator prevents these unlicensed polymerase from actually going into the gene body. So only happy polymerase make it till the end. The intron is nicely spliced and degraded within the nucleus. But in absence of integrator, what happens is that the incomplete transcribed stuff, that is to say, these intronic pieces containing the Ts, they make it into the cytoplasm, and that's how the ISR is activated and potentially causing the disease. This actually, we were able to prove that the disease were caused by looking at the cells that we got from these patients and testing if the ISR is activated in them or not. Indeed, if we look at the fibroblast, ATF4 levels are higher, the kinase that increases ATF4 level is higher, and also the sensor levels are higher. More importantly, the sensor PKR was in proximity with this double-stranded RNA, and even more striking, what we were able to see was if we correct these mutations, make these fibroblasts into iPSCs, we see the reversal, that is, integrator levels rise up. And this results in a reduction of these ISR markers, ATF4 and its sensor. In fact, if we try to differentiate these cells into neural progenitor cells, we see increased double-stranded RNA in these cells as well, leading to my final slide, where we think that this chronic dysregulated ISR, this is what contributes to the integrator-mediated disease. And this work has now recently been published at Cell, came out online a few weeks back. I encourage you to go for the details in there. But at the same time, I would like to thank all the people that made it possible from my lab, our collaborators, the core facilities, and of course, the funding agencies. I'd be happy to take questions and get your feedback on the story now. Thank you.
Thanks, Apoorva, for the wonderful presentation. If you have any questions, feel free to type it in the Q&A box, or you can raise your hand so we can unmute you to ask the question. So the first question, it's a very nice talk. Could you please explain a little bit more about the TT-seq, as in how it is different from TSS-seq?
Right. So the TT-seq is coming from the Clamor lab, and what it does is they add something called 4SU, which is an analog of uridine into the media for a very short duration, just for 10 minutes, and then this uridine makes its way into the RNA, and we can purify this RNA with these higher uridines. Now, this gives us elongation across the whole gene, whereas the TSS-seq is a very different method, which is used to identify only the start sites, whereas TT-seq is used to measure elongation, or rather nascent RNA synthesis.
And so, have you run any long-read seq to confirm that these introns are preliminary coming from premature terminated transcripts versus poorly spliced transcripts?
Yeah, that's a great suggestion. We haven't done that to prove that these are part of the intact RNA, primarily because we were not 100% sure that the secondary structure that gets formed will we be able to override it and see it in the long read sequencing. But yes, that was on our list for a very long time. Great point.
Do these integrator mutations, like at different subunits, all lead to similar phenotype, or they are very different phenotypes?
Actually, they are of a very wide phenotype. So I think with these mutations that I just showed, there were effects on other types of cells as well. But at the same time, the other mutations that have been seen, for example, in the other subunits, in those also there are certain neurological phenotypes, but they weren't so drastic as we saw here.
So I guess if they all lead to integrator complex loss, that's what you presented at the beginning, like the whole complex is gone. How do they have so dramatic different like phenotypes or different cells?
Yeah, I think it's a good question. I think it's coming from the fact that how much are the other members of the RNA processing machinery, for example, the exosome or these other nucleases which eat up the excessive RNA that is around. If it gets overwhelmed, that is when these aberrant transcripts with the repetitive sequence will make it into the cytoplasm. So maybe it's a function of how much are these other nucleases expressed in different kinds of cell, which determines what cell or what kind of phenotype will come up.
I think you have a lot of questions. We will just take one last one. So are there any phenomenon that contribute to the ISR and like the ER stress also upregulated upon integrator depletion?
That's a great question. We tested that. We tested at least for a couple of other sensors. We did a knockout on them and tried to see if we can rescue this effect. And it was only the PKR knockout that rescued the ISR activation phenotype that we see. But this was in this particular hex cell type. We cannot rule out that in some other cell type, like in the neurons, if we would make them, other kind of stress sensors, other kind of double-stranded RNA sensors would also be activated. So that experiment still needs to be done, I think.
Great. You still have tons of questions
All
in the
right.
queue. Sorry, if you don't mind, and I would really appreciate. We would really appreciate if you can type the questions in the Q&A box. And because of the time, we are going to move on to the second talk. Thank you again.
Thanks a lot to everyone who listened in, and thanks a lot to the Fragile Nucleosome community.